this video we're going to be looking at the cell cycle and specifically certain phases and starting to discuss what's happening in the cell during some of those phases. We're going to break it up into more than one video as there are multiple phases to look at. One of the first phases is interphase, and interphase is just a period of growth between cell divisions. So the cell has just got done dividing, so we have two very small cells as the cell has just divided in half, and so there's a lot of growth happening in this time. There are actually three phases that make up interphase, and we'll look at those in a second. The cell cycle, though, in and of itself, is just all of the events that the cell goes through as it grows and as it divides. So from start to finish, all the events that happen. We can break it up into four main groups. We have G1, S, and G2, and we'll talk about what each of those mean. But G1, S, and G2 are all part of interphase. We have interphase, and then we have M, and M stands for mitosis. And with mitosis, we'll have some that will break down into some multiple steps as well. So we're going to take a look at interphase right now, and interphase makes up more than 3/4 of your cell cycle. So this whole entire picture is the cell cycle, and interphase is this area that is right now in yellow, and so you see G1, S, and you see G2. So if we take a look at the events of interphase and re-mention G1, S, and G2, let's look at what happens. G1, we like to think of G1 as growth. So this is kind of the first growth phase. The cells are going to do the majority of their growing here. And keep in mind the cell was cut in half, and so they may need to make some new organelles. We definitely will need to make some new proteins. The word synthesize needs to make. So we're making organelles. We might need to make more Golgi apparatus. We might need to make more ribosomes, depending on what the cell needs to make. And then we go from G1 into the S phase, and the S phase is rather lengthy, time-wise. The S phase is where your chromosomes are being replicated, and so S is going to be standing for synthesis. That's what the S stands for. And you're synthesizing chromosomes. You're making DNA. So this is where a single chromosome will make a copy of itself, and then we get this. Remember the two sister chromatids, where these are identical halves. That is happening right here in the S phase. And lastly, for interphase, we have G2, and G2 is where the cell is about ready to divide. So it is grown, it has copied its chromosomes, so G2 is kind of the finishing touches. G is still going to stand for growth, so this is the second growth phase. So it may do a little bit of growing, but more than likely it's going to be working on developing any organelles that it might need for cell division, making sure it has enough energy to go through cell division. So it's kind of putting all the finishing touches on that before it gets ready to do quite a bit of work. There's a lot of energy and work involved in the next set of phases, which are called mitosis. Now mitosis is broken down into four individual phases, and you may hear me say PMAT. That's kind of like Roy G. Biv. It's an acronym for the names of the phases. So P for prophase, M for metaphase, A will be for anaphase, and T is for telophase. So there we have our PMAT, and those are the phases of mitosis. And remember, mitosis is where our nucleus is breaking apart. This is where our nucleus is dividing in half. So we've already looked at interphase. We're going to be looking over the next, between this video and the next one, we'll be looking at these phases of mitosis. And really mitosis is done with telophase. Cytoplasm dividing is cytokinesis. And we'll get to that one as well. So we're going to look at prophase, and then we'll end this video. But prophase is the first phase of mitosis. It's also the longest phase of mitosis. Now if we look back at this picture here, it's not the longest phase of the whole entire cell cycle. Prophase takes up this much time, where S takes up this much time. So it's not the longest phase total, but it is the longest phase of mitosis. So here are some things that are happening during prophase. First of all, your chromosomes become visible. While they're in interphase, They're in their chromatin form, which is the stringy form. They're not condensed down into chromosomes, so they're not visible to us during interphase. But during prophase, they are. Centrioles. Centrioles are something that we looked at. That's in animals only, so plant cells you wouldn't see these. But in animals, the centrioles are going to move to opposite sides of the nucleus, and I'll have some pictures in a moment that show all of these pieces. A spindle is something you're going to see during prophase, and a spindle is a piece of microtubule, and it helps to separate your chromosomes. So it's actually going to attach to chromosomes, and it will pull them apart so that they can divide in half, that the two identical pieces will get pulled away from each other. The chromosomes will go ahead and attach to those spindles. The plants we mentioned don't have centrioles, so you won't see those in a plant cell. It's one of the ways you can tell a plant cell from an animal cell. The nucleolus is going to disappear, so that dark spot inside the nucleus. Remember, the nucleus is dividing, so the nucleolus is going to temporarily disappear. It will reform toward the end. And lastly, the nuclear membrane is going to break down, so there's no nuclear membrane anymore. Essentially, the nucleus has kind of become part of the cytoplasm. Now here's a picture, and this needs to be in your notes, so you need to pause it, that's fine. I am going to be adding some labels, so please add those labels as well. But at the beginning of prophase, this would be kind of what you're seeing. So this is the nucleus, and notice that it's a dashed line, and that dash is to represent that the nuclear envelope is already starting to go apart, to disappear. Notice you can see the chromosomes, so they're in their copied shape. Nucleolus is still there, so if we're labeling things, okay, these would be your chromosomes, and we have the nucleolus. These are things that we should still know from our previous chapter. These are your centrioles. Notice they haven't split apart yet. At the beginning of prophase, everything is still kind of condensed down. This is your nuclear envelope, or nuclear membrane, depending on what it was called there on the previous slide. Now if we take a look toward the end of prophase, so time has happened. Prophase takes time. At the end of prophase, here are your centrioles. So the centrioles have moved to opposite sides of the cell. We still have our chromosomes. I won't relabel those, but we have this new object, this line here, and that's our spindle fiber. And so the spindle has attached itself one side to the centriole, one side to the chromosome. Specifically to the centromere of the chromosome is where they're going to attach. So you can just see the chromosomes are just wiggling around wherever. They're not in any pattern quite yet. But we have spindle fibers, lots of spindle fibers. No nucleus, no nuclear membrane. All of that is gone at this point in time. If you need more time, by all means pause this and keep writing, but I have one more picture, and this just is a picture I found online that just shows you a little bit of a different idea. Centrioles, the spindles coming from the centrioles. You have the nucleus, so this would be kind of early prophase because the nuclear envelope is still fully intact. By the end of prophase, that nuclear envelope is completely gone. So that's just another idea of what prophase looks like.